Hello, Jocelyn McCarthy here, Biology 225, and tonight I'm going to talk about the biochemical testing for microbial identification using different types of media. And we have the list here. It says Methyl Red, Vogue's Proskauer, and catalase testing. Just reading off this. And for the first one with the Methyl Red, we were asked to do an experiment on using the MRVP broth that were thawed out. These things. So, these broth were inoculated originally with different bacteria that were asked, and those were the E. coli and the S. epidermidis from the nutrient broth that were made in the past, in the beginning of this semester, which was November 7th. And these were inoculated as such, and it was incubated for 48 hours. After that 48-hour period, we were told to go ahead and put it in different test tubes and labeled it. Okay. They were labeled. Okay. And they were divided into 4 different test tubes because 2 of these were used with the Methyl Red, and then the other 2 were used for the Vogue's Proskauer experiment. So, with the Methyl and Red, these were the different media that were added to it. These are the Regents B, Methyl Red, and all that. I just stored mine upright into this cup with a cover to avoid spillage. With the Methyl Red experiment, it showed the acid fermentation. That's the whole point to that project, to see whether the bacteria is positive or negative. So, with that said, with the Methyl Red, it is used to test the pH of the bacteria. In this case, after adding the Methyl Red, clearly it shows reddish. So, as you notice, this is the S. epidermidis, and then this is the E. coli. The main difference is the color, maybe, but very similar. This one is a lot brighter red, and this one's sort of red tinge to it with a yellow, but it still turned positive according to the guide on the lab report. It still tested red. It still tested positive because it shows the reddish tint on both of the test tubes. Now, for the Vogue's Proskauer test, this is very similar to the Methyl Red testing. However, instead of using the Methyl Red agent that's stored in here now, we were asked to go ahead and add Regents B. First, it was Regents A. After that, we were asked to add the Regents B, and the medium were let it stand for 30 minutes and observe the color. So the turbidity, that was what I was asking for. This one, the test result would be positive also. This is because with this test, there's a lot of what is that? Condensation? Not condensation, but it is also positive. When I did that test, it's because it turned. It says here. It's fermentation. Okay, so the purpose of that Regents B is also for fermentation. And the word that we're looking for is the acetoin, which turns red in the presence of Barrett's reagent, indicating a positive result of the butanol fermentation. So I'm sorry, this test is negative because you don't see any reddish tint on this one clearly. These are all clear with the naked eye. Okay. All right, lastly is a catalase experiment. It's a catalase testing. So these were the plated agar that were inoculated for the test using the hydrogen peroxide. As we know that hydrogen peroxide is a household antiseptic liquid, just like rubbing alcohol. When you have cuts, it's recommended to put hydrogen peroxide on a cut, and it bubbles. So what that means is that there's a catalase, and it's positive. So the same thing with this experiment. We were asked to go ahead and inoculate bacteria and smeared it on these glass tubes. After smearing that, we were asked to go ahead and add drops of hydrogen peroxide onto the slide and see if it bubbles, and it did. So both bacteria are both positive because of the bubbles that I saw. And it said if the bubbles is not existent, then the result would be negative result. So the same thing, when there's a cut on a wound and putting hydrogen peroxide on it, it should bubble to let you know that there's bacteria in it, and you keep doing that until it stops bubbling as an antiseptic. And then if you want, you can clean it also with alcohol to prevent from getting contaminated as well. All right, so that concludes my video for tonight. Thank you for watching. Bye-bye.